Hello, MIP 611. My name is Kayla Crouch, and today I will be talking to you about crystallography. So there are a few methods of crystallography, and for this presentation I will be discussing X-ray crystallography specifically. This brings us to the approach. So crystallography is a method used to essentially see what the protein of interest actually looks like. Crystallography focuses on the 3D structure of proteins by using high-quality protein crystals and analyzing diffraction patterns. The methods of this approach includes having a purified protein. So prior to beginning this, you need to complete protein expression and protein purification, and it needs to be high quality. And then the next step is crystallization. Here the protein is suspended in a precipitant solution and it creates a lattice of crystals. Then we move on to X-ray crystallography. Here the crystallized protein will be subjected to X-rays that form a narrow amplitude, and this allows us to view the diffraction using a detector. And the diffracted image will then be processed and an electron density map will be generated, giving an atomic model of the protein. The next step is validation. Here the model is checked by different validation procedures to basically assess the quality and overall fit to the experimental data. And then you'll be able to use the protein for whatever application you're completing this for. And then there's a few controls in this method, but for time's sake, I will be discussing the positive and negative. So a positive control for this method includes using the same method for your specific purified protein, but instead using a well-known characterized protein. And then the results should match that, ensuring that the results are accurate and reliable. And another control is a negative control, and this is using a crystal that is known to have no diffraction produced and ensuring the outcomes match this, and this helps eliminate experimental errors. This brings us to our connections, advantages, and limitations. For connections, this relates back to our Module 4 protein expression and analysis material. In Lesson 4.1, we discussed how crystallography can be used to figure out what the protein of interest actually looks like, specifically using crystallography as a way to map our protein correctly. In Lesson 4.3, we discussed it again as a way to aid in using purified recombinant proteins to study protein, RNA, and DNA interactions as well. Advantages. Advantages of this approach includes having the ability to produce high-resolution detailed information about the 3D arrangement of the crystal structure of our proteins. And this approach offers a much higher resolution compared to other techniques such as the cryo-EM. Another advantage is that this is widely used in a well-established method. This process has a large applicability. This method is relatively cheap compared to other processes. And disadvantages include that this method does require crystallization, obviously, whereas other methods like the cryo-EM does not. And so this can be challenging when working with specific proteins that can be resistant to crystallization, and overall it can be a time-consuming process. Another disadvantage is that there can be damage to the protein through X-ray radiation damage, and another disadvantage is that you need a larger sample amount compared to the cryo-EM methods. The first primary manuscript I'll be presenting is titled Protein Production in E. coli for Structural Studies by X-ray Crystallography. In this paper, the authors are working to find strategies to overcome two big issues in X-ray crystallography, which include protein solubility and crystallization. This figure shows the ribbon diagrams of 8 solved structures, And I included this because it shows a variety of different structures that are and can be produced using X-ray crystallography. The next manuscript is titled Characterizations of Aptamer Protein Complexes by X-ray Crystallography and Alternative Approaches. In this paper, the authors are trying to analyze the crystallization of the aptamer protein complex and review technical issues of using X-ray crystallography with this process, while also providing details of the specific protein interactions. Figure A shows the crystal structure of the thrombin binding complex, and through the structure they were able to visualize the core of the thrombin binding aptamers, which are the two stacked G complexes shown in the gold of Figure A. And Figure B just shows this bound to exosite 1. Lastly, this primary manuscript is titled Protein Crystallization for X-ray Crystallography. Like I mentioned before, crystallization is typically referred to as the bottleneck of crystallography, and in this paper the authors are providing two different experimental approaches to crystallization for crystallography. If we look at the figure, this figure was very cool to me. I included it because it demonstrates what the typical outcomes of a protein crystallization experiment are. If we look at A represents amorphous precipitation, and this results when the protein or precipitant are high in concentration. If we look at B represents phase separation. The protein or the detergent can separate into different phases when mixed with precipitants at a high concentration. C represents rod-shaped crystal proteins. These are specifically from ATCSN7 obtained in polyethylene glycol 8000 and magnesium acetate. D represents lysosome crystals, and these crystals were obtained in 1 mole of NaCl and sodium acetate with a pH of 4.9. And E shows an unsaturated clear drop. And these are my references. If you have any questions or comments, please let me know below. And thank you for listening.